So, first question, as usual, I'm watching comment section. First question, the second messengers A mediate the intracellular response to many different hormones and neurotransmitters. B are substances that interact with first messengers outside the cell. And C, they are substances that bind to first messengers in the cell membrane. And then D, they are found in the brain. E, they are hormones secreted by cells in response to stimulation by another hormone. Which of those options is accurate? Second messengers. Yes, Cecilia says B. Tionge says B. And the rest of us? Ngulani says C. Alright, anyone else with a different perspective? Okay, so um, let me find the slides on G coupled receptors. I'm sure that's what we talked about. That's what we talked about second messengers. Okay, okay, so this is where second messengers should be. Okay, so this is the definition. The first messenger, a chemical messenger that comes from outside of the cell, the ECF, and activates a receptor. Okay, I'll repeat that. First messenger is a chemical messenger that comes from outside of the cell and activates a receptor. And this is usually a hormone. Okay, and then the second messenger comes into the cell, e.g., Ca2 plus, during muscle contraction, or are produced inside the cell due to activation of the receptor. And examples are cyclic AMP, cyclic GMP, inositol triphosphate, diacylglycerol, DAG, and Ca2 plus itself. So, where I wanted us to concentrate is this G protein coupled receptor illustration here. Okay, so the second messenger in this case is cyclic AMP. Okay, cyclic AMP, and it's produced by adenine cyclase. It's produced inside the cell. So other second messengers like CA2 plus can come out of from outside the cell from the ECF like CA2 plus itself. So CA2 plus in the neuromuscular junction, for example, is going to come from the ECF, come into the cell, then it's going to induce release of SCH vesicles from the presynaptic membrane, of course. So those are the two examples that you can use when referring to these second messengers. So from that background, we can now assess the options uh, that we have here concerning. Okay, right. So we are saying that the second messengers. We're saying that second messengers A mediate the intracellular response to many different hormones and neurotransmitters. Then the options that I'm seeing in the inbox are substances that interact with first messengers outside the cell. Okay, so this is not accurate because there's basically no interaction between the first messenger and the second messenger. Don't I don't really see, or rather, there is what? Please. Mhm. Okay. Essentials. I hope essentials has. Usa bright. Tumira bright. Suelta. Okay. Ya fez ya fez ya mega fez mas talvez sim. Seria um pouco essa pessoa não sair. That's right. All right, I can't find the text in this book, so I'll be able to um, send it through once I find it. All right, question. We can move on to question 11. In normal human blood, A, there are more white blood cells than red blood cells. B, the iron is mostly in hemoglobin. Sorry. C, there are more lymphocytes than neutrophils. D, the eosinophil is the most common type of white blood cell. And then E, there are more platelets than red blood cells. There are more platelets than red blood cells. Okay, there are more lymphocytes. There are more. Bulani says C. There are more lymphocytes than neutrophils. Anyone else? Celia says B. The iron is mostly in hemoglobin. So the correct answer is indeed B. The iron is mostly in hemoglobin. The iron is mostly in hemoglobin. Okay, the rest of these are not. The yes, the rest are not. Alright, so it's, saying 11, that's the correct answer B. Move on to 12. Initiation of an action potential in skeletal muscle. A requires the release of norepinephrine. B requires the release of acetylcholine. C requires the spatial facilitation. And then D requires the temporal facilitation. And E is inhibited by high concentration of calcium Ca2 plus at the neuromuscular junction. Zero says B requires release of acetylcholine. That is accurate. Release of acetylcholine cause an action potential at the for the skeletal muscles. 13. Which of the following physiological processes is not correctly paired with the structure? A. End plate potential, muscarinic cholinergic receptor. B. Action potential generation, initial segment. And C. Electrical transmission, gap junction. D. Negative feedback inhibition, the Renshaw cell. And E. Synaptic vesicle docking and fusion, presynaptic nerve terminal. Okay, B. An action potential generation initial segment. Please note the question says which of the following is not correctly paired. So we are looking for one that is not correctly paired. And B is correctly paired. So we can remove B from what we've already answered. So let's try again. The next minute. Okay, E. Extruder says E. Synaptic vesicle docking and fusion, presynaptic nerve terminal. Cecilia says A. Okay, so the accurate one is A. The end plate potential and the muscarinic cholinergic receptor. That is wrong. So Cecilia, where exactly is the end plate potential initiated if this is not the correct structure? You can type it in quickly. Okay, so for end plate potential, what you need rather is nicotinic receptors, not muscarinic receptors. Yeah, I hope you're okay with that one. That's what is wrong. So muscarinic is not correct. It's supposed to be nicotinic receptors. Action potential generation, axon hillock, that is accurate. Electrical transmission, gap junction, that is correct. Gap junction, we know that we have connexins connecting two cells together, and those allow transfer of materials directly between the two neighboring cells. And then one of those materials that can be transferred across that gap junction is the uh, the ions, the ionic properties, not ionic properties, the ionic components of the cells. These are the ions like the calciums themselves, the sodiums themselves, sodium ions. So due to that, you can transfer an electrical uh, impulse directly between two cells because of that connection of the gap junction. So that is true. D. Negative feedback inhibition, the Renshaw cell. This is one of the interneurons that is found in the central nervous system. And that interneuron is particularly an inhibitory interneuron. That means once the impulse goes through that cell, it's going to cause inhibition, and it's usually in the negative feedback loops of most neurological pathways. All right, and then E says synaptic vesicle docking and fusion. This happens in the presynaptic nerve terminal. That is accurate because the presynaptic nerve terminal is the one that contains the synaptic vessels. So the synaptic vesicle docking refers to that process where the synaptic vesicle fuses with the presynaptic nerve membrane so that it can release its contents into the synaptic cleft. So the synaptic vesicle has to dock and fuse with the presynaptic nerve membrane, and then it's going to release its contents into the synaptic cleft. I hope that is okay. So I can show you a picture quickly on that one.
Kasih Jombe, kita cek bagaimana? Sekarang saja nak kita kacai, kemana kacai?
All right, so I was talking about the thing that should come in mind is this picture there that shows you that is the vesicle there, the synaptic vesicle fusing with the cellular membrane, and once it fuses, the SCH that is inside there is going to be released into the synaptic cleft. That is basically that, and then a cholinergic receptor, nicotinic receptor in this case is going to so this one, if it's a synapse, not a neuromuscular junction, you can have a cholinergic receptor like a what's that one that was in the option that we were just talking about? Yes, that one. You know it when you see it. So after the impulse transmission through the synapse, the SCH will be broken down into choline and acetate by acetylcholinesterase, SCHE. And they usually ask this Kama question as well. So do not forget that part. So this is a presynaptic membrane, nerve membrane, and then this is the postsynaptic. So that arrangement should tell you where the impulse is flowing and how it's flowing. All right, we can go back to yes. So which of the following is true about circulating erythrocytes? A. Travel at a slower velocity in venules than in capillaries. B. Remain unchanged as they pass through the capillaries. C. They are distributed evenly across the bloodstream in a large blood vessel. And D. They may show an intracellular network pattern if appropriately stained. E. They are about 1% nucleated. True concerning circulating erythrocytes. So concerning circulating erythrocytes, true option here is option D. They may show an intracellular network pattern if appropriately stained. This option directly refers to the reticulocytes, so you can see that meshwork pattern of the reticular fibers which contain even some Is it RNA or DNA fragments? Okay, so these other options are wrong. Travel at a slower velocity in venules and capillaries. The opposite. They travel slower in the capillaries than in the venules because of the cross-section areas. Venules are bigger than capillaries, so obviously, just because of that difference in cross-section area, you're going to have a slower velocity in the capillaries. And they remain unchanged as they pass through the capillaries. Of course, they're going to be disfigured a bit because of the diameter of the capillaries. It's very small. It's very, very small. So they're going to be the membrane is going to have to deform a bit to accommodate that small diameter. Distributed evenly across the bloodstream in large blood vessels, due to uh, factors of the way the blood flows in large blood vessels, laminar flow and everything. Find that they are not distributed equally. They will gravitate towards more to the center than in the periphery. All right, they're about 1% nucleated. It's less than 1%. So this option is not true. About 1% means around, but the true form of this option is they are less than 1% nucleated. Question 15. The thick myofilaments are mostly made of A. Myosin. Okay, this question is too straightforward. Myosin, B. Myoglobin, C. Troponin, D. Tropomyosin, E. Actin. Okay, let's not waste time here. This is straightforward. A. The functions of tropomyosin in skeletal muscles include A. Acting as a relaxing protein at rest by covering up the sites where myosin binds to actin. B. Generating ATP, which is passed to the contractile mechanism. C. Sliding on actin to produce shortening. And D. Releasing Ca2 plus after initiation of contraction. E. Binding to myosin during contraction. What is the true concerning the functions of tropomyosin in skeletal muscles? Okay, that is that it acts as a relaxing protein by covering up the sites where the myosin will bind to actin. That is accurate. That is basically what tropomyosin does. So don't forget, I think I put something related to this. So you have your actin filaments there arranged like that. Those round things. We assume they are actin filaments. And tropomyosin is this band that is going around like that. That's the tropomyosin, and there is a troponin complex on that as well. There's troponin I, troponin C, and troponin T, whose functions are illustrated here, and we did go over them as well. So if you need to remember these, you can go back to these slides. They're available on the drive. Yeah. All right. We're almost done. Almost done. Question 17. This paper only had 20 MCQs, so we're going to end there today. What is a motor unit? A motor unit. A single muscle fiber and all of the motor neurons that innervate it. A single muscle fiber and all of the motor neurons that innervate it. B. All the muscles that affect the movement of any given joint. C. All of the motor neurons supplying a single muscle. D. A single motor neuron and all the muscle fibers it innervates. And E. Two motor neurons supplying a single muscle. Okay, Kumbilani says D. Celia, that is accurate. So a single motor neuron and all of the muscle fibers that it innervates, you call that collective a motor unit. And question 18. Regarding the skeletal muscle, the ryanodine receptor is located in the A, the SR membrane, the SR terminal systems, B, sarcolemma, D, Seca pump. Okay, I've missed options. C, Seca pump, G, tubule, E, the cytosol. Blind says A, yes, that is accurate. The SR terminal systems, that's where you have the ryanodine receptors, and they are coupled to um, uh, DHP receptors. Yes. All right. Question 19. The cross bridges of the sarcomere in skeletal muscle are made up of. These questions are not. These questions are too direct. They don't have any physiology in them. This is anatomy. This is a question that you should be asked in anatomy. Cross bridges of the sarcomere in skeletal muscle are made up of A, tropomyosin, myelin, actin, myosin, or troponin. Cross bridges. Okay, so cross bridges, will find them on myosin. Please go check out that picture of. This is just swattering a picture. It's just swattering a picture, and you know where cross bridge is and what it looks like. Then it ends there. No explanation, nothing. Just supposed to know how it looks like, what it is. Stimulation of an inhibitory presynaptic fiber causes A, the release of gamma aminobutyric acid or GABA, B, this determination of excitatory postsynaptic potential in the postsynaptic neuron, C, the secretion of acetylcholine, D, the release of norepinephrine, and E, initiation of an inhibitory postsynaptic potential in the postsynaptic neuron. Stimulation of an inhibitory presynaptic fiber. Cecilia says B, determination of an excitatory postsynaptic potential in the postsynaptic neuron. With a different perspective. Okay, so the correct answer is rather A, the release of GABA. Okay, so this one is talking about the presynaptic fiber. So if the presynaptic fiber is inhibitory, what it's going to release, because you know a presynaptic fiber always has to release contents from its neurotransmitter vesicles. So if it's an inhibitory, it's going to have an inhibitory neurotransmitter in the vesicles. If it's stimulatory, it's going to have a stimulatory or excitatory neurotransmitter in its vesicles. So for what we've been talking about, this normal synapse, which assumes that the impulses are traveling across that synapse, that is excitatory. So ACH, for example, is an excitatory neurotransmitter. But if an inhibitory presynaptic fiber is stimulated, it's going to cause release of gamma aminobutyric acid. For example, in the Renshaw cell, that one we talked about. So GABA will be produced so that it causes hyperpolarization in the postsynaptic membrane. So the postsynaptic membrane will be unable to produce an action potential because GABA makes it to be hyperpolarized instead of the normal depolarization. Okay, are we following so far? Are we following so far? All right. Okay, question 21, the last question for today. A 47-year-old woman was admitted to the hospital after experiencing nausea, vomiting for about 2 days, followed by severe muscle weakness and neurologic symptoms, including that word that I don't know how to pronounce it, and dysphagia. She indicated she had eaten at a restaurant in the evening before the symptoms began. Lab tests were positive for Clostridium botulinum, that word. Okay, so biomedical scientists will bring this person to your lab and then find that this is what you find. You'll be finding a lot of these in pathology next year. So the question, the neurotoxins A exert all their adverse effects by acting centrally rather than peripherally. B, such as tetanus. So if we're reading this option, say the neurotoxins exert all their adverse effects by acting centrally rather than peripherally. Then the neurotoxins such as tetanus toxin bind reversibly to the supposed to be presynaptic membrane at the neurotransmitter or rather the neuromuscular junction. And then option C, if the neurotoxins such as botulinum toxin prevent the release of acetylcholine from motor neurons due to cleavage of either synaptosome associated proteins or vesicle associated membrane proteins. This question is very long. Then option D, the neurotoxins block the reuptake of neurotransmitters into presynaptic terminals. Then the neurotoxins reach the soma of the neuron by diffusion into the spinal cord. What did you put as the answer there? Okay, so for this one, whenever you hear botulinum toxin, just think of preventing the release of acetylcholine from motor neurons. That's all you're gonna have to swatter from there. The rest of this is just excess data. Yeah, just information to support the story. But botulinum toxin prevents the release of acetylcholine from motor neurons. So, however they ask you this question, just know that fact and you'll be able to navigate it. Alright, that is it for today. Unless there is any question, let's meet in the evening. Biomed and pharmacy. I don't know if red and physio is what your timetable says, but you should have a session tonight as well. And pharma and physio, we have anatomy tonight, the usual time. Let's see how far we can push that paper we started yesterday. Yes, we need to at least maybe do 85% of it, so that the remaining part. And you were supposed to get an assignment. You're getting it today. The one for